Insect Transgenesis today. Some of this, the first little bit of it will be a little bit of review, and then we'll go into some of the more insect-specific stuff that you need to talk about, and we will talk about some different experimental strategies that people are often using in insect transgenesis, in fruit flies and in other insects. Okay, so there's three things that you need for insect transgenesis. One is a mechanism of insertion. So that means like the molecular molecules, like the enzymes. If you have a chromosome and you want to put something on it, you need to have the enzymes that can catalyze that reaction. If that's recombination or if that's transposon insertion, you need to have a mechanism of insertion. Okay, you need a mode of delivery. So that means you need a vehicle in which to get these enzymes that you need and the thing that you're going to insert. You need a vehicle to deliver that to the insect. Okay, and maybe that's going to be like the insect body, or maybe that's the insect egg, depending on different contexts, but you need a mode of delivery. So we'll talk about different modes of delivery. And then just like any bacterial transformation or yeast transformation, you need a selective marker. But as you noted, as you learned, selective markers in bacteria tend to be a little different in yeast, and they tend to be a little different in insects and in plants. So there's different selective markers and different selective strategies. So in bacteria, our selective markers are usually what? Yeah, they're antibiotics. And we give them like resistance cassettes, like can resistance, chloramphenicol resistance, AMP resistance, and we add antibiotics. And then the yeast selective markers are what? Yeah, I think we call that auxotrophic markers. So we make a deletion, so they're missing something. And then in our plasmids, we usually encode that gene that codes for that deletion, like UR3. And then we leave uracil out of the media. So they select for that plasmid. So in insects, most commonly what we do is we do things that cause like phenotypes that you can see. Phenotype, so visual phenotypes. So one example of this, most famous example is normally in fruit flies, actually I should color these in, wild type fruit flies have red eyes, okay? And kind of like yeast, what we do is the flies that we make transgenic flies from, we actually use mutant lines that are called W lines. They have white eyes. So they have a mutation in the gene that codes for whatever chemical it is that makes the eyes red. That's been mutated, and so they're now white. And then oftentimes what we do is, so we take these lines, and if we want to add in gene X into these lines, into these flies, we put it on a plasmid. I'll talk about a little bit about different plasmids. Let's just say PUSP, ATTB. Let's say we put gene X right here in our plasmid. It's got a promoter, and the selective marker is what's called mini white. You'll just see it as W. Looks like that. And mini white encodes the gene that makes red eyes. So in Drosophila, everything is opposite of what you would expect. So when they name genes, oftentimes they discover that, they discover that gene based on a process of mutagenesis. So somebody, when they first identified the white gene, they took wild type fruit flies and they mutagenized them. And then they pulled out a white-eyed mutant, and then they named that fly, this is the white-eyed fly. And then when they discovered the gene that was mutated, they named that the white gene. But if you have a wild-type white gene, your eyes are what color? They're red, yeah. So if you have a mutant white gene, your eyes are white. So we complement, this is called complementation. we complement that mutation that when we insert our gene X from this plasmid, we're also inserting this cassette that's a mini white gene, and then the eyes become red. So in flies, let's say we get 100 eggs and we inject 100 of them, and let's say 50% die, and let's say 50% survive. And of those ones that survive, maybe only 1 to 5 out of 100 come out with red eyes. And then we just take those ones that have red eyes and we put those in a little tube and we just pass those red-eyed flies. So that's how we select for transgenes in Drosophila. I'll expand this a little bit. I'll talk about more in mosquitoes and other things like that. So one of the downsides of this is in Drosophila, this is easy because it's all been optimized and worked out for us by other people, and there's all kinds of white-eyed lines. But mosquitoes, you can get white-eyed lines, but it's not as common that you want to work with a model line of mosquitoes. If you're making a transgenic mosquito, usually you're trying to do something that's very applied. So oftentimes you want to work on a strain that's very closely related to the strain in the field. So if you want to, say, make a transgenic mosquito that you're going to release in Brazil, oftentimes you want to make it from that Brazilian strain. So oftentimes you don't have access to like a white-eyed mutant, even though you could theoretically do it that way. So oftentimes what we do is plasmids that we would insert a transgene into a mosquito for, they'll often have what's called a reporter. Very similar concept, but it's a little bit different, where we put like GFP, and if we want to insert, say, gene X, we put a mosquito promoter in front of GFP. Oftentimes they'll do it in eyes. Oftentimes they'll do like an eye promoter. So that means it's a gene that normally turns on in the eyes. And then when this plasmid gets inserted into the mosquito chromosome, the eye promoter will cause the eyes to glow green when you shine a light on it. So oftentimes we'll select our transgenic mosquitoes by you microinject a bunch of eggs, you put the eggs in the water, they'll hatch, they form these larvae that look like this. And you can just screen the larvae based on if they have green eyes or not. If they don't have green eyes, you throw them away. If they do, then you keep them. And you just keep selecting that way. So it's like visual markers are what we're often using in insects for transgenesis. Let's see. Okay, so let's talk about that point 1, the mechanisms of insertion. And this will be a little bit of review, but I want to talk about it more in detail. And now I'm going to expect you guys to kind of like know this stuff pretty well. So for like this stuff would be on like a test. So one of the means is transposon insertion. And I think we talked about this, actually we did. We already talked about this a little bit in the context of Drosophila. So you need to remember that with transposon-based insertion, you need 2 plasmids. What are the plasmids called? Yeah, either the helper is the suicide one. There's the helper and then the donor. And what's the unique difference about these 2 plasmids? What do they have on them?
The helper has...
Is it restriction on one side, or like they have the same sequences on both ends of the insert?
That's kind of cool. You're kind of close. So in the helper encodes a transposase. Remember the jumping genes. The helper encodes, so it's an enzyme. It encodes a gene. It's a transposase. It's got a promoter. Oftentimes, it's usually a constitutive promoter, which means what? It's always on. So oftentimes you'll see like ubiquitin or actin. These are things that are always on, strongly expressed. in front of this transposase. And as soon as you microinject that, the central dogma, this promoter will turn on this gene. It'll get made into messenger RNA, which will get made into a protein. So this protein is the transposase. And the transposase is a DNA binding protein. And it recognizes sites that are on the donor that flank, that it's been designed so that they flank your gene that you want to insert. And the transposase will grab and cut your gene out. And then it can paste into the chromosome. So the transposon insertion is the cut and paste mechanism. Cut and paste. Does that make sense? That good? Okay. We talked about that a little bit. Got that. And we talked about the downsides. What are the downsides of this means of insertion?
It's random where it gets placed.
It's random. Random insertion. What are the other downsides? No, we don't have to worry about that. That's going to be when you're building constructs and like ligating stuff. This is after we've already built these constructs.
You have to make 2 plasmids.
Yeah, that's a downside. You gotta deliver 2 things. You gotta deliver 2 components. And there are ways that simplify that. So that's a good point. The other thing is that you get, because it's random, you get what are called positional effects. What are positional effects?
It can mess up the gene if it gets inserted into the
Yeah, that would be one. So let's say you have a chromosome, you have a bunch of genes on this chromosome, and if your transposon inserts right there in the middle of your gene, you've now broken that gene. And if that gene encodes something important, then you have no idea if that's affecting your downstream experiments that you're going to do. So that can be a problem. That's one positional effect. There's more than one different type of positional effect. So another positional effect is, we talked a little bit about differences between eucs and procs. What does the prokaryotic or the eukaryotic DNA look like? How's it different? It's in chromosomes. Well, prokaryotes have chromosomes. They have circular chromosomes. But I see what you're saying. There's multiple chromosomes. But what's structurally different about those chromosomes? It's larger, and so that means they have to do something to it.
You have to unfold it.
They got to package it. It needs to be packaged. So when you look at the eukaryotic DNA, it's coated with these histone proteins. The beads on a string, they form the 30 nanometer fiber, which condenses. So when you look at a eukaryotic chromosome, there's regions of it that are called heterochromatin, and euchromatin. What do these words mean? Yeah, heterochromatin is compact. it's assembled onto the nucleosomes. It's not open for transcription. It's not open. So it's not being actively transcribed. So you can get transposed. Let's say this is a region of heterochromatin right here. You can get your transposon to insert in there. So let's say you inserted your gene X in a region of heterochromatin. Do you think it's going to express well or poorly? It's going to express very poorly because everything that's around it is compacted and it's not going to be accessible from the RNA polymerase. You can get also the reverse effect. So let's say this whole chromosome, let's say it's all heterochromosomes except right here, this region right here. Okay? And you get your other gene to insert right here in a region of euchromatin. Now ask the same question. Is that transgene going to be expressed poorly or really well? Really well. And maybe you don't want it expressed really well. Like maybe too much will actually kill the organism. So these are 2 different types of positional effects where you can't necessarily have complete control of expression of your transgene unless you know where the insertion site is, unless you know the site. Okay? So that's a severe limitation of the transposon insertion mechanism. But there are ways that we can use this to study things. So let's talk about a couple of these different strategies. Let's talk about the enhancer trap. This is a type of experiment. What is an enhancer? Yeah, it's a region that will enhance transcription. Enhance, spell that wrong, transcription. And does it have to be close or far away, or where do you find these? You can find them downstream, you can find them upstream. Oftentimes you can find them very, very, very far away because they loop around. And you might have, so you might have something that's 20,000, 100,000 base pairs away from your actual gene, but because of the structure of the chromatin, it might always be in physical proximity to the gene of interest. So you could find an enhancer right here that might do something to regulate transcription of gene X. Okay, so oftentimes we want to know where these enhancers, where these enhancer regions that regulate transcription of genes, where they are, where you find them. So let me ask the question. If you have a new organism, a new Drosophila, and you want to map where enhancers are, how could you do that? What's a strategy you could use? You want to find out where all the enhancers are on all the chromosomes. So one thing you could do is, let's say you do a transposon-based strategy. So you have your helper. Your helper has your transposase. I'll expect you to know this. What the name of the transposase that people are most commonly using in Drosophila, the way that we first transformed insects, was by using the P element transposase. So I want you to know P element. That's a key word you should know. That's the type of transposase that is used in Drosophila. Okay, so we took P element helpers. So the P element helper encodes the P element, the P transposase. And then we have our donor. And we can put a reporter in the donor. So let's say a reporter would be GFP. And we can make a whole bunch of different flies because it's random insertion. Let's say we make 1,000 flies that are randomly inserted into the chromosomes of fruit flies. And then we can just look, we can shine a light on all these fruit flies, and we can see which ones glow green. And we can see which ones glow the most green, or the least green. And then we can map the sites of insertion. And then we can correlate the expression levels of the same gene. It's all GFP. But the only difference is the positional effect, right? The only difference is the insertion. So if we, basically what I'm telling you is if we insert GFPs at different sites in different flies on different chromosomes, we can identify regions of DNA that are expressed very highly or very lowly, and then we can make a conclusion that somewhere in that area, some kind of an enhancer. That's called an enhancer trap. Does that make sense? So that's a famous style of experiment that utilizes the randomness as a screening method to learn something. What was the second thing? Oh, okay. The other thing I want to talk about is there's another way to utilize the randomness. Randomness can be a good thing because you can use it in screens. So what's another way? So describe to me how a mutagenesis strategy might work with. How could you conceive of an experiment to use transposon mutagenesis to learn something? let's say you want to study wing development. How could you learn something about that using transposons and mutagenesis?
You could try to stick transposons near the genes that code for wings.
Yeah, so let's say you don't even know the genes that code for wings, but you want to learn what they are. You just want to study wing development. So what you could do is you just have a normal, here you don't even need two. You do not need two plasmids in this strategy. You just need the normal transposase, and it's going to have sites for itself. You just microinject this, if there's a promoter, it'll get made, it'll grab its own gene, and it'll randomly insert to different sites. And then what are you going to select for? What are you going to look for?
If its wings are broken.
Yeah, you're going to look for weird wings. Weird wings. So there's a very famous Nobel Prize winning experiment where the guy, his name is Ed Lewis, I think. He discovered Hox genes. What are Hox genes? We'll talk about this in insect development. So there's actually genes that encode for, like, you turn that gene on, it builds a wing. Like it starts building a wing if it gets turned on. These are Hox genes. So the way that the guy discovered this is, well, he didn't use a transposase. He used chemical mutagenesis, but you could use this same strategy, and I'll just talk about it in theory. So what you would do is you could just microinject thousands of eggs with transposons, and then as they grow up, the ones that survive, you don't care about any of them unless their wings are messed up. So you look for abnormal wings. So your selection is actually like wing mutants. And Ed Lewis, so he did this with a chemical, I think EMS. So this is a chemical that induces mutations. Same thing as a transposon. If the transposon inserts in a gene, it can break genes, cause mutations. Okay. And what he found is he found fruit flies. So fruit flies are dipterans. What's diptera mean? Has two. So dips two wings, means two wings. Do all insects have two wings? No, the ancestral insect wings are four. Ancestrally, insects have four. So if you look at like dragonflies or Ephemeroptera, which is, what is Ephemeroptera? I'm forgetting. But if you look at like ancestral wings, the ancestral wing formation is you have four wings. And in Diptera, they've lost two of their wings. And the two wings have been mutated into little nubs that are called halteres. These are like little gyroscopes that help them fly. So all dipterans, mosquitoes, fruit flies, dipterans have two wings. And this guy, Ed Lewis, identified the gene that causes wings to be created because he made a mutant, and there was a mutant fruit fly that came out that instead of halteres had four wings. So he found by mutating just one single gene, a Hox gene, you could get that fly to make a whole new set of wings where the halteres should be. So it's just one gene that turns on that controls, it says build a wing, and it tells the fly to build a wing. Okay, so that's one strategy that you could use with using transposons to study like insect development, or to learn which genes do what by just trying to find mutants, make a bunch of mutants. That's called, this is called reverse genetics. Why is it called reverse genetics?
You're mutating it back to an ancestral form.
You don't know what you're looking for. You're screening for a phenotype. And then you find a phenotype and you trace it back to the gene that was mutated. So what's forward genetics? Forward genetics is you want to, you know the gene, and you mutate that gene specifically on purpose, you make a mutant, and then you study the phenotype. That's forward genetics. This is reverse genetics. So a lot of people do different combinations of reverse and forward genetics with insect transgenesis. Okay, so let me ask you a question. We know all this stuff about P elements in Drosophila. Okay. Will what we know in Drosophila work in mosquitoes? Can we use P elements in mosquitoes? Maybe. You can use it in other fruit flies. So there's one agricultural pest, D. Suzuki'i. P elements might work in D. Suzuki'i. But you can't take P elements and have them work in mosquitoes or moths or other things. It doesn't work like that. So a lot of these transposons are very specific for their organism. Okay? They're very, very specific. So, okay, let me elaborate on that. Let's say but you don't care about fruit flies. You need to make a mosquito mutant. How could you do it? How could you find a transposon that would work in mosquito?
Look for a natural virus.
Very good. Yeah, not a virus, but yeah, you look at the genome. You could look at the genome of mosquitoes, and you could try to find, there'd be thousands of them. You just find a transposon in the mosquito genome, and you just make sure you check that catalytic sites. What are the catalytic sites? What's that mean?
How the enzyme works.
Yeah, how the enzyme works. You have to study the enzyme. Transposons are going to have to cut DNA because they have to cut and grab DNA, put it somewhere else, cut and paste. So you got to make a lot of these are DDE residues. You make sure they have those conserved because a lot of times you'll find broken transposons, not active ones. But if you find an active one, let me ask a question. How would you find an active one? How would you know if it was active or not?
You just try it.
You could try it. Yeah, you'd have to try it. That would be your last, but you wouldn't want to check. You'd want to check other things before you spend the time to try it. Because to try it, you'd have to build all these constructs. What else would you want to check? So I told you the first thing you'd want to check is you'd want to check catalytic sites. And you'd want to check the literature. What else could you look for? What would you expect to see if a transposon was active? Do you think there'd be a lot of copies of it or just one copy of it? Jumping genes, things that jump around. Yeah, you find a lot of them. So a lot of times if you are finding an active transposon, you might see 100 copies of it. It jumped from one chromosome and say it got it, say the mom had it, and then they mated and the dad also had it, and it jumped from the mom's chromosome to the dad's chromosome. Now all of a sudden there's 2 copies in the dad's. Does that make sense? And then let's say the next generation, that 2 copy in the dad's chromosome mates with another mom that has 1, and it jumps from the mom's to the dad's. Now that dad has 3 copies of it. That accumulates. So in active transposons, you'll find hundreds of copies of these things, exactly the same gene, all over the genome. Okay? And then you could make the assumption, now that one is worth trying out because there's 100 copies of it in the cell. It looks like it's active. So let's clone that out. Let's clone that gene out. Let's build our own donor and our own helper. So donor, helper, I drew this reversed. So in the helper, we would clone that one that we want to try. We would probably change its promoter, and we'd have to look for its special site, and we could make our own donor vector, insert gene X, and then, yeah, we could try to study whether or not that one would work in mosquitoes, okay? So lots of people have done this. There's many different types of transposons that work for different kinds of insects. So there are, in mosquitoes, you can use one called MOS1, which is a Mariner type transposon. You don't need to know that. You don't need to know the name, but I'm just telling you that there's different ones. There's one called piggyback. There's one called hobo. There's one called, what are the other ones? Hermes. These are all special ones that people have identified that have different contexts. They work in some insects, they don't work in other insects. Okay. So there's insect specificity for transposons. There's one called Sleeping Beauty. This one's story's cool. So in Sleeping Beauty, they found Sleeping Beauty in fish. Let's say this is the gene, and they noticed that it was broken. So that means it didn't have it was either disrupted or it had mutations, and they cloned it out and they repaired it. So I've taught you all the techniques you would need to know to do this. You would clone out the gene with your primers, put it in a plasmid, okay? And then let's say it had a frame shift mutation. It has premature stop codon. How could you correct that? Yeah, you use like a quick change. Your quick change or a slim, you fix it. Now it's perfect. And then now you can do the test, and they did the test, and it was an awesome transposon for zebrafish. So zebrafish are a model for development because they're see-through. They're see-through fish. You can see the organs develop. And people use now Sleeping Beauty to make mutants in zebrafish. And they came from a broken transposon, which they repaired, and now they use it as a biotechnology tool. So this is, again, why now you guys know, you know everything you would need to know to recreate that entire study. Let's see. Okay, I already told you a little bit of this story, so I'll just tell it quick again, just to reiterate. So people have now corrected, we don't use random transposons unless you have a specific reason to do the randomness. We use PhiC31 Integrase, which is site-specific. And I told you how Cohen-Vanken did that. I told you that he made a helper with P, so it's the P element. And he made an insertion, a suicide plasmid, a donor, with a viral integration site, ATT, it's either P or B, I'll just say P. And then he made 1,000 different lines where this ATTP landing site got inserted at different points on the chromosomes, just like that enhancer trap thing. And now we have 1,000 different lines you can choose from, and you can use PhiC31 Integrase, which is the enzyme, to integrate your gene X, as long as it has the airplane site, ATTP, and if ATT, well, it's the opposite, B or P. If that matches and finds the ATTB site, your PhiC31 will organize a recombination event to insert your gene at a site-specific spot. So let me elaborate on this strategy a little bit more, because I told you that before. You guys kind of remember that? Okay, so let me ask a question, because let's go into this a little bit more detail. How do you deliver the PhiC31 integrase? How do you deliver that?
Like a recombination.
How do you deliver the enzyme? You need two components for this to work. You need the PhiC31 integrase, and you need your donor. So how do you get that protein there?
I guess you'd inject it.
Inject what?
You'd have to make some.
Okay, so this is good. There's multiple ways you could do this. If you were making it, you'd have to make it from what? Plasma, right? So one way you could do this, you could do it like a standard transposon. You could do it like a donor. So you would have a helper, and this time the helper would encode the PhiC31 integrase. And you could do it with a donor. So you give the helper and the donor would just have the ATTP, okay? But that's not much easier. It's site-specific, but it's still kind of, you've got to deliver two things. Another way you could do it is, just like you said, you could make an E. coli expression plasmid. You could have E. coli make the PhiC31 protein, maybe with like a his tag, and you purify that protein, and then you could mix the protein plus the donor plasmid with your ATTP, and then you could microinject protein plus plasmid. That could also work, but that's still a lot of pain because it's a lot of work to purify proteins, right? Is there any easier way in which you could conceive of you doing this? Well, what if you integrated the PhiC31 gene straight into the Drosophila genome itself? then that eliminates your need to deliver it. So what people have done is they made a fly that has white eyes, and in its chromosome, they have inserted a transgene. That transgene is PhiC31 Integrase. Where do they want that integrase to express? What would the promoter look like?
Be near the one for the eyes, so you can tell if they change.
But then you're only going to think about what happens. It's a good thing that you say that, but let me point out the flaw in the logic there. Let's say you have it expressing in the eyes, PhiC31 integrase. Where are you going to get integrations of your donor? In the eyes or in the body? In the eyes. And then this thing mates. Is it going to pass on an insertion in its eyeballs to its next generation? No. So where do you want this thing to express? The eggs. So they're going to have a germline promoter, which turns on the PhiC31 integrase in the eggs. And then you collect eggs from this. It's got white eyes. Collect its eggs. And in each one of those eggs, it's already going to be making PhiC31 integrase. And all you need to do is inject your donor plasmid with a needle into each of these. Whatever hatches up and has red eyes got your transgenic insertion. Does that make sense? And it's going to be, it won't always be, but it's most likely to be heritable. So that means if this is the F0, you cross a red eye to a red eye, and the F1s should still have red eyes if the transgene inserted into the germline. And then you can just maintain that line. Does that make sense? So this relates to CRISPR. We won't go too much into CRISPR right now because I haven't given a full lecture on all the components, but I'll just quickly outline it. In CRISPR, you need two components, just like this. You need a Cas9, which is the enzyme, and you need guide RNAs, which tell the enzyme where to cut. So people are making transgenic fruit flies that have an endogenous Cas9, and what would the promoter for that be? Yeah, it'd be the same thing. It'd be a germline promoter of the Cas9, and then all you need to inject is the guide RNAs. Makes it a little bit easier. You don't have to go through the process of purifying the Cas9 enzyme, although sometimes that can be more efficient. So it depends on the context. But there's sometimes when you want, sometimes it's a lot easier to have one of these components kind of taken care of already by the fly or by the mosquito. Okay, let's talk about the microinjection process. So you collect eggs. Let's say you put a bunch of fruit flies in a cup. They lay their eggs, and you collect them. Is it going to be better to inject the embryos early or later? Early. Why? Why does timing matter?
But you want your gene to get onto the chromosome before they start making lots of copies.
Is this like mosaic? Like you want
You're both kind of right.
You want to divide. You want as many cells to have what you want in it.
Yes.
So you don't get mosaic.
Right. So you're both kind of right. Let me explain a little bit more. Let's do an example. Let's say you have two eggs. This egg is 0 minutes development. It was literally just fertilized. This one is 2 hours developed. It's got cells. It's got a mesoderm, endoderm, ectoderm. I'll do a whole lecture on insect development to sort of explain all this. It's already starting to get organs. It's get stuff inside, and the eyes might already be formed. The gonads might already be formed. You inject here, and you inject here, here, if you get the insertion at that first division, every single cell in this organism that grows up is going to have that insertion because it's all derived from that first embryo. Does that make sense? You inject here, you might only get an insertion right here. Maybe these things form, maybe these cells right here form wings. You're only going to have your insertion in the wings. And then when this thing grows up and when it makes a baby, the insertion isn't going to be here in the gonads where you need it. It's going to be in the wings. It's not going to get passed on. So this is what's going to be called a mosaic, where not all the cells are the same thing. These cells have the insertion, but everything else doesn't. If you hit it at the very first division, it's not a mosaic. Everything has it. You can still work with a mosaic if you inject at the right spot. Let's say you inject right there and you hit the cells that become the ovaries. Then you're good. You're golden because you got your insertion in the ovaries. Even though it's a little bit later, you're still good. But you always want to do it earlier than later because of this reason. Does that make sense? So this is the dichotomy of somatic versus germline. What's somatic tissue? Yeah, body cells. Mutation is useless if it's in the body because we want things that will be passed on to the next generation. You can't study mosaics. Well, I mean, you can, but it's a lot weaker experimentally to study a mosaic because it's not consistent. Every experiment you need to try to control for as many variables as possible. And if you can't even have control over how many cells in the body have your insertion, that already shoots yourself in the leg. Okay, so one of the reasons that insects are awesome for transgenic experiments, fruit flies, mosquitoes, is because we can synchronize egg laying. Okay, so we can put a whole bunch of flies in a little cup with an apple juice plate, and they won't lay any eggs until we add some yeast. The yeast makes them go crazy. They mate, and then they lay thousands of eggs. And we can collect all these eggs, and we can inject them right away. So we can hit all the eggs very early. Take a cow. Is a cow egg going to be easier or harder to study than a bunch of fruit fly eggs? It's going to be a lot harder because we don't have as much control over that fertilization process. And that cow might only fertilize one egg. And maybe you have to pull out the egg and maybe you got to fertilize it yourself in like a test tube and then you got to like implant it. It's a lot more complicated. Same thing with so let's examine like a mosquito. So a mosquito, you blood feed them. You feed them blood. Okay. The mosquitoes, females, take the blood. They digest the blood meal and they make eggs. So now you got a pregnant female ready to lay eggs after 48 hours of you giving her a blood meal. She'll hold on to those eggs. She won't fertilize them yet until if it's Aedes aegypti, you shut off the lights, and you give them some water in a cup. So if this is a cup full of water, Aedes aegypti likes to lay its eggs right on the surface of water. So you give it a little cup full of water with a little piece of paper, you shut off the lights, that female will then start laying eggs and she fertilizes them as she's laying them. So she takes an egg, she gives it some sperm. She already mated, so she's got a little organ in there called the spermatheca, which holds sperm. And as she's pumping out eggs, she'll fertilize them herself with the sperm. And then she'll lay these fertilized eggs and you can come back in 20 minutes. You can take these eggs, 100 eggs now, and you can microinject 100 of these eggs. Does that make sense? So this is called synchronous egg laying. So insects are amazing for transgenesis because we can collect so many eggs and we can inject them very, very quickly. Okay, so let's talk about the injection. So we're collecting eggs. What's the difference between a chicken egg and a human egg?
Chicken egg is much larger.
Yes. What about the structure of the surface?
Has like a hard outer
Chicken eggs have a hard shell, okay? And insect eggs are the same way. So animals that have external development, like chickens, where the eggs are growing up outside of the body, that's not like a womb, it's not like a mammal, where the embryo grows inside of you. Insects are the same way, most insects. The development is all outside of the body. So they have a hard eggshell to protect that embryo. Okay? So when you microinject an insect egg, one of the problems is there is this hard eggshell called the chorion, and it's very hard. So you have to make needles. Let's say you make a needle that looks like this, a needle that looks like this, and a needle that looks like this. Which one of these is going to be the best?
The bottom,
Why?
the top one wouldn't go through. I feel like the
Wilson
middle
is right. Let me explain why. Say you take this one, you stab this egg. It'll work. You'll still be able to pierce it. You'll stab it. But then you've created a massive hole in that egg, and then you pull it away.
The V shape will allow the outer shell to fold back down. You know, it's just not a hole left. Just like
Yeah.
the
So-
middle one would just leave like a little pierce hole with the
Well, let me talk about this one.
pull it down.
So hang on. So you're right. This one's going to create a giant hole. And as soon as you pull that needle out, everything's just going to spill out and it's just going to die. This one, what's going to happen is you're going to try to pierce that eggshell and you won't be able to pierce, and your needle will go like this. It'll bend and it'll snap. You won't actually even be able to pierce the chorion with this type of needle. This one has the benefits of both. It's super sharp, okay? And it's strong because it's thicker. So what people will do when they pull needles is they'll pull them. They'll pull them like this, and then they'll sharpen them with a bevel. They'll actually like take a little microscopic needle and put it on a little like stone, sharpening stone that spins. And then they'll use a microscope to like get it down and then sharpen it. And then you sharpen it, and then you can make a tiny hole. You poke, you inject your substance, your two plasmids, and then you can pull away. And the membrane can just come back together and seal itself. And then these things will survive. So actually the construction of the needle is very important in these processes. So this is important for mosquitoes. Mosquitoes are aquatic. When we inject them, we can't remove their eggshells. But not all insects are the same. So when we inject Drosophila, one of the things we do is we do what's called decorionation. Corionation. What's the chorion? The chorion is the outer shell. So decorionation is actually removing the eggshell. So if you take Drosophila embryos and you put them in bleach, it will strip off the eggshell. And then you can use a very, very, very thin needle, because then you're just piercing a membrane. And then the survival rate is going to be higher in Drosophila because you're making it just a tiny, tiny hole. Okay? But that only works with Drosophila, because Drosophila is a land animal. It's better at surviving. It's better at keeping in, its embryos are better at keeping in water. Mosquitoes, do they lay their eggs by the water or on the land? Yeah, they're aquatic. So they need to be kept wet. So when you remove the eggshell of the mosquito, most of those embryos will just die because they'll just dry out too quickly. So depending on the biology of the organism, you need to look at the biology of the eggs, and there's different ways you can optimize the protocol for making things work depending on the biology of the species. So that's a very important consideration. Let's see. What else we're talking about? I think that's it. You guys have any questions? Okay. So that's insect transgenesis. Next time on Wednesday, we'll do insect development. And then I'll go back to the beginning of the module and do plant transgenesis and some of the other stuff.
What is this capillary for?
Oh, that's how you make the needle. So you get a machine, which is called a needle puller. And a capillary is a very, very thin glass tube. And you put these glass tubes in the machine, and there's a hot filament, and there's a spring-loaded thing that pulls this way and this way. You press a button, and this filament heats up, and it pulls the glass, and it pulls it, and then it pulls it slow at first, and then it pulls it real quick and snaps it. And then that's how you produce needles.
So they're glass?
They're glass. They're made of glass. These are you call them microcapillaries, or I guess microinjection needles. Yeah, they're made of glass. All right, good work. So we